Good morning, children. Are you ready for revising reproduction in plants, the first chapter in biology? Yes? Okay. So, you know what is reproduction. Reproduction is essential feature for all living organisms on the earth. Okay, and especially plant reproduction is not only important for its own survival but also for the survival and continuation of the existence of other living organisms, either directly or indirectly. And we know that reproduction is mainly divided into two categories: asexual reproduction and sexual reproduction. So under asexual reproduction, you have learnt about reproduction in lower plants, reproduction in higher plants. And under sexual reproduction, we have learnt about pre-fertilization structure and events, fertilization and post-fertilization structure and events. The reproduction method which helps to continue its own species without the involvement of gametes is called as asexual reproduction. Reproduction in lower plants, we have formation of conidia. Conidia are asexually produced spores that are borne externally to the cells that produce them. As you can see here, an example of the penicillium. Yes, and then we have budding. Budding, in this hydra plant, you have the picture here, how the bud, it is detached from the parent plant. And then we learn about fragmentation. If you can look at the example there for fragmentation in Spirogyra. Picture here, production of gemma. Here the single cell or the modified bud, it detaches from the parent and develops to grow into a new individual. Then we have regeneration. Regeneration is where it detaches its tail end and each half of the organism, it develops, regenerates into a new living organism. And then we have binary fission. Binary fission, in this process an organism duplicates its genetic material, the DNA, and then divides into different parts. Moving on to reproduction in higher plants, we have natural methods and artificial methods. Under natural methods, you have learnt about vegetative reproduction in root. The roots of some plants develop vegetative or adventitious buds on them. Then vegetative reproduction in stem. The axillary buds from the nodes of rhizome, as you see in the picture. Okay, the eye of the tuber give rise to new plants. And then we have vegetative reproduction in leaf. Here in some plants, adventitious buds are developed on their leaves. When they detach from the parent plant, they grow into new individual plants, as you see in the example of Bryophyllum. Artificial methods, we have used the conventional methods, three types: cutting, grafting and layering. So first we talk about the cutting. Okay, it is the method of producing a new plant by cutting the plant parts such as root, stem and leaf from the parent plant. Okay, the cut part is placed in a suitable medium for growth. Depending upon the part used, it is called as root cutting, stem cutting or leaf cutting. So you have an example of stem cutting here, hibiscus. Okay, and then we move on to grafting. Grafting, here in this, parts of two different plants are joined so that they continue to grow as one plant. That is grafting. And we have learnt about bud grafting, approach grafting, okay, and tongue grafting, crown grafting, wedge grafting. Next is that the layering, mound layering. Look at this carefully. Here, if you see, this method is applied for the plants having flexible branches. So, the lower branch with the leaves is bent to the ground and part of the stem is buried in the soil and tip of the branch is exposed to this above soil. After the root emerges from the part of the stem buried in the soil, a cut is made from the parent plant so that the buried part grow into a separate new plant. Layering. What happens in this method? The stem is girdled at the nodal region and hormones are applied to this region which promotes rooting, and then this portion is covered with damp or moist soil using a polythene sheet and root emerges from that portion. We now move on to sexual reproduction in plants. Here if you see, the sexual reproduction involves the production and fusion of male and female gametes. The former is called as gametogenesis and the later process is the fertilization. So under sexual reproduction, we will now first start with pre-fertilization structure and events, then about the fertilization and then post-fertilization structure and events. Gametic fusion is of three types: isogamy, anisogamy and oogamy. Okay. For example, in algae external fertilization takes place, whereas in higher plants, internal fertilization occurs. Okay, so look at the flower. Flower, for a morphologist, a flower is a highly condensed shoot meant for reproduction. A flower possesses 4 whorls: calyx, corolla, androecium and gynoecium. Okay, androecium and gynoecium are essential organs. The process or changes involved in sexual reproduction of higher plants, they include 3 stages as we just now saw. Okay, so pre-fertilization structure and events, here it is the first thing that we'll be learning, you learned is the male reproductive part, androecium. Okay, androecium is made up of stamens. Each stamen possesses an anther and a filament as you see in the picture. Okay, then we see the anther bears pollen grains which represents the male gametophyte. Look at this, the blue part is the male part. Okay, anther and the filament, and all those in a pink color, stigma, style, ovary and ovules, they are the female parts in the flower. Okay, then we see about the development of anther. A very young anther develops as a homogeneous mass of cells surrounded by an epidermis. In this picture, if you see, the stages involved in the formation of haploid microspores from diploid microspore mother cell through meiosis is called as microsporogenesis. Microspores and pollen grains. Microspores are the immediate product of meiosis of the microspore mother cell, whereas the pollen grain is derived from the microspores. Okay, so in the development of male gametophyte, the microspore is the first cell of the male gametophyte and it is haploid. Haploid is the name given to a cell which contains only one set of chromosomes, that is from a single parent. The development of male gametophyte takes place while they are still in the microsporangium. The nucleus of the microspore divides mitotically to form a vegetative and a generative nucleus. So have a look at this picture and then we learn about the stages in the development of male gametophyte. So you start with exine, intine, nucleus and germ, pore. Then you have the vacuole and the nucleus. Then nucleus is divided we have vegetative cell and the generative cell. And then finally we have male gametes and tube nucleus. So the androecium is the sum of the male reproductive organs and gynoecium is the sum of the female reproductive organs. So the outermost whorl of the flower has green leafy structures known as sepals. The ovule, it is also called as megasporangium, and it is protected by one or two coverings called as integuments. And the types of ovules that we see here, they are classified based on their orientation, form and position of the micropyle with respect to the funicle and the chalaza. Okay, so the 6 types are orthotropous, anatropous, hemianatropous, campylotropous, amphitropous and cercinotropous. So look at this picture. Here you have microsporogenesis and megasporogenesis. Okay, so the process of development of a megaspore from a megaspore mother cell is called as megasporogenesis. Development of ovule and embryo sac. The embryo sac develops from one megaspore and is called monosporic embryo sac. The nucleus of functional megaspore divides by 3 mitotic divisions to form 8 nuclei as you see in this picture. The 8 nuclei are organized into antipodals, egg apparatus and polar nuclei as in polygonum type of embryo sac. Pollination. Pollination, as we know, it is a mechanism which provides food, shelter, etc. for the pollinating animals. And many plants are pollinated by a particular animal species and the flowers are modified accordingly. And thus there we see co-evolution between the plants and the animals. So under pollination you have self-pollination or autogamy and cross-pollination. Under self-pollination we have cleistogamy, which is flowers never open and expose the reproductive organs and thus pollination is carried out within the closed flower. Okay, and then we have homogamy. When the stamens and the stigma in a flower, they mature at the same time, then we call it as homogamy. Then incomplete dichogamy, where the flower and the stamen, the stamen and the stigma of a flower mature at different times. Sometimes the time of maturation of these essential organs overlap so that it becomes favorable for self-pollination. And under cross-pollination, geitonogamy, it is when the pollen deposits on another flower of the same individual plant. It is said to be geitonogamy. And xenogamy is when the pollen genetically different deposits on another flower of a different plant of the same species. Then that is called a xenogamy. Okay. Contrivances of cross-pollination. First one is decliny or unisexuality. And under that, monoecious, male and female flowers on the same plant like maize. And dioecious, male and female flowers on different plants. Okay. Monocliny or bisexuality, you have dichogamy. First one. Under dichogamy is bisexual flowers, anthers and stigmas mature at different times and it checks self-pollination. Dichogamy is of 2 types, protandry and protogeny. And hercogamy is In bisexual flowers, the essential organs, the stamens and the stigmas are arranged in such a way that self-pollination becomes impossible. Okay, so we then learn about the heterostyli. In heterostyli is where some plants produce 2 or 3 different forms of flowers that are different in their length of the stamens and the style. Pollination will take place only between organs of the same length. So distyli is plant which produces 2 forms of flowers, and tristyli is where plants produce 3 kinds of flowers with respect to the length of the style and stamens. Then we see here pollen from flowers of one type can pollinate only with the other 2 types but not of their own type. Okay. Finally, we have self-sterility or self-incompatibility. What does this mean? When the pollen grain of a flower reaches the stigma of the same, it is unable to germinate or it is prevented to germinate on its own stigma. Okay, so it is a genetic mechanism that we see here. We then move on to agents of pollination. We have under pollination we have self-pollination and cross-pollination. So under cross-pollination we have abiotic agencies and biotic agencies. Abiotic agencies, first one is anemophily, where wind is used for pollination, and such plants are called as anemophilous plants. Okay, hydrophily, it is where water is used for pollination. It is by water, pollination by water, and the flowers pollinated by water are called as hydrophilous plants. In hydrophily, you have 2 more divisions like epihydrophily. Epihydrophily is pollination occurs at the water level, like as in Vallisneria spiralis. And in hypohydrophily, here pollination occurs inside the water as in Ceratophyllum. Under biotic agencies, we have zoophily. Pollination by the agency of animals is called as zoophily. Okay, and the flowers which are pollinated this way, those flowers are called as zoophilous. Under zoophily, we have ornithophily. Ornithophily is pollination by birds. And cheiropterophily, where pollination is carried out by bats. Malacophily. Malacophily is pollination by slugs and snails. Okay. And entomophily, it is pollination by insects. Pollination by ant, we have learnt many times, it is myrmecophily. Okay. Then we move on to fertilization. Under sexual reproduction, we have fertilization. Plant fertilization means it is the joining of the male and female gametes which results in the zygote, as you can see in this picture. Okay, the most generalized form of this process requires 4 steps: pollination, germination, penetration of the ovule, and then the fertilization. After fertilization, the zygote divides to form an embryo. So, here the different stages involved in double fertilization. They are germination of pollen to form the pollen tube in the stigma. Then growth of the pollen tube in the style. Then we have the direction of pollen tube towards the micropyle of the ovule. Entry of the pollen tube into one of the synagogues of the embryo sac. Discharge of male gametes, syngamy and triple fusion. Okay, so the events from pollen deposition on the stigma to the entry of pollen tube into the ovule, it is called as pollen pistil interaction. Here we have post-fertilization changes in the flower of an angiosperm. Path before fertilization, how they are transformed after fertilization. The sepals, petals, stamen, style and stigma are there, and after fertilization usually they wither and fall off. The ovary is now the fruit, ovule is the seed, egg becomes the zygote, funicle is stalk of the seed. Okay, and thus there are lots of transformation that happens after fertilization. Then we have move on to post fertilization structure and events. The events after fertilization are endosperm, embryo development, formation of seed, fruits, okay, and they are called as post-fertilization changes. So, the primary endosperm nucleus, PEN, divides immediately after fertilization but before the zygote starts to divide to form the endosperm. And depending upon the mode of development, there are three types of endosperms. Nuclear endosperm, as you see in the picture, then the cellular endosperm and the helobial endosperm. Finally, we also have ruminate endosperm. We then move on to the development of dicot embryo. The embryo develops at micropylar end of the embryonic sac, as you see in the picture. The zygote undergoes transverse division to form upper or terminal cell and lower or basal cell. Further divisions in the zygote during the development, it leads to the formation of embryo. Embryo undergoes globular heart-shaped changes, as you see the picture, before reaching a mature stage. The mature embryo has a radicle, okay, two cotyledons and a plumule. Then about the seed. The fertilized ovule is called as seed and it possesses an embryo, endosperm and a protective coat. Seeds may be endospermous, okay, and non-endospermous, like as in beans and mango, right? The mature seeds are attached to the fruit wall by a stalk called funiculus. The outer coat is called testa, and the inner coat, the thin membranous part, is called as the tegmen. The portion of embryonal axis projecting beyond the cotyledons is called as the radicle or the embryonic root. The other end of the axis is called as the embryonic shoot, which is the plumule. So, a quick recap of all that we learned today. Reproduction, asexual, sexual. Under asexual, lower plants, we learnt about the formation of conidia, budding, fragmentation, production of gemma, regeneration and binary fission. And under higher plants, we had natural methods and artificial methods. Natural methods, vegetative reproduction in root, stem and leaf. And artificial methods, we had conventional methods like cutting, grafting and layering. and we learnt about sexual reproduction, pre-fertilization structure and events, then about fertilization, finally about the post-fertilization structure and events. Happy learning, children. Thank you.